Let's go over topic 2.8. Let's start with the word diffusion. So we already know diffusion is when we have our molecules going from high concentration to low concentration. Now we're going to bring this to osmosis. Okay, so if the substance that's being diffused is water, then we're going to call it osmosis. Okay, so that process is known as osmosis. So we looked at that in our notes when we did notes on 2.6 and 2.7, that if the molecule is dissolving a gas, that is diffusion. But if we're referring to water, then it's osmosis. Now, in comparison, both osmosis and diffusion, your final result in both of them is going to be an equal solute concentration on both sides. Now, the key differences that we're going to see between osmosis and diffusion is that in osmosis, the membrane is usually only permeable to water and not solute. Okay, so that's the key thing for us to remember. In diffusion, on the other hand, our membrane is usually permeable to solutes. Okay, so that's our biggest, the key that you need to remember is that when we're looking at those comparisons in diffusion, solutes can go through. In osmosis, the only thing going through is water. Now, similar to diffusion, when we're looking at osmosis, our water is going to want to go from the high concentration to low concentration. Okay, so this is very similar. It's going to want to flow from high to low. Now, we want to look at this in terms of our solute. When we have less solute, that means a watery solution. Whereas when we have more solute, that's going to be our concentrated solution. So the water is going to flow from less solute to more solute. Okay, because we're comparing that, we know when we're referred to solute, that means that we're adding it to the amount of water. So that less solute means that we have more water, and more solute means we have less water. So the water is still flowing from high concentration to low concentration. But if they're referring to the phrasing of solute, you need to think of what that means for the water concentration. Now, we also want to keep in mind that anytime we're referring to water, this is not equal to just pure water. Okay, because of the fact that water makes such a good solvent, we're usually going to have water exist as a solution. Okay, just the way that it's able to dissolve everything and be a universal solvent means that you will usually find it as a solution that does have something dissolved. Now remember the word solution. That's where we have our liquid solvent, such as water, with our solute added to it. Now let's talk about tonicity. Okay, when we're referring to tonicity, this is what we're using to describe osmotic gradients. Okay, so we're looking at a comparison. So when you're going to use these terms, they need to be used in comparison to something else. So for example, the most common way you're going to see it is going to be comparison of a cell to its environment or the solution that it's placed in. All right, let's start with isotonic. Okay, isotonic. So when we're using the term isotonic, we're using it when something is isotonic in comparison to something else. Okay, so the term iso means same. So in an isotonic comparison, both the solute concentrations are going to be equal. So just keep that in mind, that iso means same, and we're referring to tonic, we're referring to the strength, or like the concentration. Now, if we were referring to an isotonic solution, so this is where our solution is what we are referring to in comparison to something else. The solute concentration of the solution is going to equal the solute concentration of our cell. Okay, so if we are comparing our solution to the cell in an isotonic solution, there will be the equal amount of solute inside the cell as the amount of solute outside the cell. Now this will mean that water is going to consistently diffuse in and out at an equal rate. Now because it's moving in and out at an equal rate, there will be no net change in the size of the cell. Now let's look over at our hypotonic. Okay, so in hypotonic solution, so I'm going to go ahead and start referring to it as just our solution, because that is the example and the phrasing that you guys are going to see in questioning, is that they are going to refer you to the solutions. So in an hypotonic solution, the word hypo means under or beneath. Okay, so think about that with our isotonic. Iso meant same, hypo means beneath. So the solute concentration of our solution is going to be less. Okay, so this will be lower. And the solute concentration in the cell will be higher. So if you were comparing, you would see that there's more solute inside of the cell and there's less solute outside of the cell. So this means that the water will flow into the cell. Okay, so we're going to see that flow of water going into the cell. And when we have this water flow into the cell, this is going to cause the cell to swell or burst, depending on the type of cell we're looking at, and we'll come back to this in a little bit. Now, I want us to kind of take a minute and wrap our minds around this concept, because this could be confusing because of the way that we are referring to it. We're referring to solute. More solute means that we have less water. Remember, solute's taking place of the water. So less solute equals more water. Okay, so the water is flowing from the area that has the higher water concentration to the area that has the lower concentration. So when we said that the solute concentration inside the cell is higher, keep that in mind, that means that water concentration is lower. Now let's move on to hypertonic. Okay, so our hypertonic solution, the word hyper is over. Okay, so it means over. So the solute concentration in a hypertonic solution, in the solution, is going to be higher, and in the cell it's going to be lower. So when we would look at our cell and compare it to its environment, you're going to see a lot more solute outside the cell and less solute inside the cell. This means that the water will flow out of your cell. Okay, so remember that less solute means that there's more space for water. We have a higher water concentration inside. And so the water is flowing out. This is going to lead the cell to shrink. Now I want us to take some time and talk about osmosis and plant cells. So we know the structure of plant cells is different from animal cells in that plant cells have cell walls. Okay, so this is going to play a role in osmosis and how we're going to look at what's happening in the cell with our tonicity. All right, so the cell wall in plant cells is a very essential component for plants because it protects plants against osmotic changes. Okay, so plants can't constantly go through different environments. They can go through a time where they would have a lot of water, or they have a time where they have a lot less water. So they need this added protection against these osmotic changes. So the cell wall is not going to change its size. It allows the cell to not change its size. Now, plant cells do have cell membranes. So when we have osmotic changes, those membranes are going to change. So those membranes will move either away from the cell wall or push up against the cell wall, depending on what's happening with the water, if the water's coming in or if the water is moving out. So cell membranes are going to be the ones that will move with the amount of water in plant cells. So the change we see with our cell membrane is what's going to reflect the osmotic changes that plant cell is going through. So I want us to take some time and look at each of our examples for iso, hypo, and hyper, and look at what we would see if we looked at plant cells. So in an isotonic solution, a plant cell is going to be normal. Okay, so the solute concentration outside and inside is the same, and we refer to this as flaccid. Okay, so the term for plant cells in isotonic is flaccid. Now let's look at hypotonic. In a hypotonic environment, we're going to see that water is coming into the cell. So what that's going to do is it's going to push that membrane all the way up against that cell wall. Okay, so we'll just see that the membrane is being pushed up against the cell wall. That is added pressure for that cell. Now the added pressure from that water on the cell wall is going to be known as turgid. So keep in hypotonic solutions, the cell is taking that water in, and taking that water in is increasing the size of its vacuole, membranes pushing out against the cell wall, and we're going to know that this is turgid. Now, turgid pressure in plant cells give them their crispiness. So think of turgid pressure and think of crisp celery. So crisp celery, that crispness is coming from the turgid pressure inside those plant cells. Now let's take a look at hypertonic. So we already know that in a hypertonic solution, the water is leaving the cell. So what we're going to see in a hypertonic solution with our plant cell, as the water leaves, the membrane will shrivel up away from the cell wall. Okay, so remember that the cell wall is keeping the plant cell from changing its size. So the size of the plant cell is not changing. What we're just seeing is that the membrane is shrinking away from the wall. And we are going to know that this is going to be called plasmolyze. Okay, so this process is where we would say that it's gone through plasmolysis. So keep those three terms in mind when it comes to our plant cells and looking at tonicity. Now let's go over to our calculations. So water potential is shown using our term psi. So I always like to think of this, you see that the symbol for water potential looks like a trident, and that trident makes you think of water. So that's our symbol for water potential. Water potential allows us to determine the direction that water is going to flow. Okay, so we know that just like diffusion, water is going to flow from an area of high concentration to low concentration. So high water potential to low water potential. Okay. And the units that we're going to see with our water potential is going to be bars. The formula for water potential is right here. We have psi equals psi of S plus psi of pressure. Okay, so pressure potential, think of what's going on. As we keep adding water into the cell, the pressure that's being put on the cell membrane is increasing. Okay, so as that increases the pressure potential. Okay, so pressure potential is going up as the amount of water going into the cell is going up. Now, our values for pressure potential are usually going to be positive. Now, another thing that we can keep in mind, if they tell you that the container is open, that means that your pressure potential is 0, since there's no pressure being applied with the container being open. And I'll come back and I'll add that in when we do a sample problem. So now let's look at solute potential. Now, what we need to understand about solute potential, if you're adding solute, that means less room for water. Therefore, our solute potential is going down. Okay, so adding our solute, we have less room for water, so the solute potential is going down. Okay, high concentrations of solute are going to equal lower solute potential. Low concentration of solutes are going to equal higher solute potential. Okay, and I know this is something you have to grasp and wrap your mind around, since it's phrased so strangely. Now, calculating solute potential, the formula is negative I CRT. I is the ionization constant. For NaCl, this is going to be 2, and for sugars such as sucrose and glucose, it's going to be 1. C is equal to concentration, so that's your molarity, and the formula for that, remember, would be just moles over liter, and that's the units as well. Pressure, so R is your pressure constant, which means it's always 0.0831. T is your temperature. Now it does need to be in Kelvin, and so you would have to convert Celsius to Kelvin by adding 273. So let's look at a practice problem. So this is a very commonly used lab in AP Biology where you're studying vegetable cores, and you're going to put them in sucrose solutions, and you're going to measure the percent change, or measure the change in mass in the vegetable after having been left in sucrose for 24 hours. So the idea behind it, the concept, is that we're going to have osmosis happening within those cores over the span of the 24 hours. Now, when we measure the mass after the 24 hours, we're going to see whether water entered or left that core. Okay, so you're going to be able to use those percentage changes in the mass and compare the different molarities you used in your experiment. Now, the ideal way for us to figure out the solute amount in the vegetable cores is to figure out what our isotonic solution was. If we figure out the molarity at which we have no change, so when the mass does not change, at that molarity of our sucrose solution, that molarity will be the same for our vegetable core solution. Okay, so this is the basic concept behind this. You end up plotting your points on a graph using your percent change, and using that, you're going to look at where did we not have a change. So you would look at the 0 on the x-axis and figure out the molarity. So of course you're probably not going to have an exact molarity that you don't see a change in your actual experiment, but by plotting the points on a graph, you can determine the molarity that would be isotonic. So they've given you data, and we're looking at zucchini cores. So zucchini cores that are placed in sucrose solutions at 27 degrees Celsius resulted in the following percent changes after 24 hours. So we're going to take these values that they gave us and we are going to plot them on the graph. So we have above 0 our increase in percent, and then below 0 we've got our decrease. So we're going to plot these points on to the graph. And then we're going to go ahead and we're going to connect our points on that graph. Okay, so plot your points for each solution and connect those points on that graph. And so when we go and we look at our actual question, we're going to be able to use this graph for our calculation. So one of the questions they can ask you is, what is the molar concentration of the solute within the zucchini cells? So to find that, you're going to look at the x-axis intercept. Okay, so the curve and best fit is going to show us the molar concentration of solute in our zucchini cells, or whatever substance that we're looking at the x-intercept. So take the spot on the x-intercept, go down and see what that molarity is. So this is approximately, let's say, 0.36 molar, okay? So this is if you need to figure out what your concentration is when they've given you the data, and you can plot it and figure that concentration out. So the formula for solute potential is -I CRT. Now we can go ahead and use what we have and calculate solute potential. I is going to be 1 because we're looking at sucrose. C is what we just figured out using the chart, 0.36 moles over liters. R is always going to be that constant for pressure, so 0.0831. And I'm running the units out so you guys can, cross out your units and see how you're left with your bars. And T is our temperature in Kelvin. So in the question, they told us that we have 27 degrees Celsius, add 273, so that's 300 Kelvin. We go ahead and we multiply it, and remember that we have that negative in our formula, so this is negative 8.97 bars. So our solute potential is negative 8.97 bars. Now let's go ahead and take this number, and we're now going to calculate our water potential. So when you're calculating the water potential, Remember that the formula for calculating water potential was solute potential plus pressure potential. So when we're calculating our water potential of the solutes within the zucchini course, we're going to take the value that we just calculated and we're going to plug it into the formula. So we know that our solute potential was negative 8.97 bars. Now it was in an open container. So remember, I mentioned this earlier, so I'm going to add this into the notes now. If we're looking at an open container, the pressure potential is going to be 0. So if you plug that into your formula, we have our solute potential there, so we can now put our pressure potential as 0. Okay, so negative 8.97 plus 0 is equal to negative 8.97. So our water potential for this example would also be negative 8.97. All right, so that is it for topic 2.8. Okay, so we did an example of each of our equations. Keep in mind when we're looking at our solute potential, your concentration can either be given to you, you can get it from graphing your data, you can also calculate it using the formula for molarity, which is moles of solute divided by the liters of your solution. Your pressure constant it's a constant, so it's 0.0831, and your temperature has to go into the formula in Kelvin. If you plug in Celsius, it's not going to work and your answer will be wrong. So you have to convert to Kelvin from Celsius. Ionization constant, these are the two that you're really going to need to know. NaCl is 2, sugars are 1. This formula altogether is available for you on the AP formula chart, and so is the formula for water potential. So use those formula charts. Okay, that is it for topic 2.8.